Dr. Menge received his BA in zoology from the University of Minnesota and a PhD in zoology from the University of Washington. Currently, Dr. Menge is a distinguished professor of zoology and the Wayne and Gladys Valley Endowed Professor of Marine Biology at Oregon State University. Dr. Menge's research has had a tremendous influence on the field of ecology. Did this turn off? On the field of ecology. Well, anyway. Dr. Menge's research has had a tremendous influence on the field of ecology, and we are honored to now include him as part of the EIS tradition. He has authored over 125 peer-reviewed publications appearing in journals such as Science, Nature, and PNAS, among many others. He is recognized as a highly cited author by ISI and is among the top 0.5% of the most cited ecologists over the past 2 decades. In recognition of his achievements in the field, he has received the George Mercer Award, a John Simon Guggenheim Fellowship, an honorary Doctor of Science degree from Long Island University, and the Western Society of Naturalists Lifetime Achievement Award. Dr. Menge's research focuses on the structure and dynamics of rocky intertidal communities, with a particular focus on linkages between ecosystems in zones of coastal upwelling. Dr. Menge's work is firmly rooted in small-scale experimental ecology, but he also seeks to understand large-scale patterns through a comparative experimental approach. As lead PI in the Partnership for Interdisciplinary Studies of Coastal Ecosystems, he is also interested in the impacts of climate change on ecological dynamics in marine communities and the application of this knowledge to the practice of marine reserves and sustainable management of marine resources. Please join me in welcoming Dr. Bruce Menge.
Is it on? No. Is it on now? Nope. Hello. It worked? Okay. So I should have a headphone. I could be like a rock star or something. Hmm? Oh. I'd probably better not do that. Okay, well, thanks very much. Whoa. It's really a pleasure to be here. I've never been to College Station, and it's really only my 3rd visit to the state of Texas. As large as it is, that's kind of amazing. I'm a marine ecologist, as was mentioned. When I got the invitation from Dan, I thought there'd been a mistake because you guys are landlocked here. There aren't really too many oceans close by. So I decided to take advantage of the mistake and accept the invitation, which I appreciated very much. Okay, so what I want to talk about today, this is something, it's sort of a Monty Python thing, now for something completely different. This is very different from what you've just heard. Although I think it's related to more to what Gary has said than it would appear at first. So I want to first of all set the stage. Because you're mostly a non-marine crowd, I need to try to explain things fairly clearly and give some background in what I'm about to talk about from a marine perspective. So following decades of observations in all of ecology, actually, but in marine ecology as well. In the 1950s, marine ecologists started to use experiments in the field to examine species interactions, and I'm referring to the experiments of Joe Connell, which sort of touched off an experimental explosion. It was delayed in some habitats, but eventually has pervaded all of the major habitats in the world. And the initial focus was usually on a single site, in other words, at a local scale. But the generality of these results, Connell's results were very spectacular. They showed strong interactions. Payne's results showed strong interactions. But their generality was unclear because typically they were done at just a single site. And that's not a criticism, that is just a statement of the resources available and the activity levels possible in those times. But eventually, people began using a multi-site approach, which I've called the comparative experimental approach, which gives you a much greater spatial perspective and allows you to address generality, at least in space. And when this was done, it was found that actually the strength of interactions would vary with environmental variation. And a particular one in marine systems, at least shallow water ones, was wave activity, which it was learned that that had a strong impact on how strong interactions were. Now, in this kind of work, the null hypothesis in all cases was that the impact of oceanography, of the coastal ocean, was essentially null or not very important, with the exception of wave disturbance. This changed in the 1980s when some marine ecologists started looking at the input of recruits to populations and pointing out that under some conditions this could be a very strong factor influencing the abundance of populations and thereby of community structure. So this forced a shift from the local to larger scales because larval dispersal is driven by larger scale sorts of processes. And then a final step was made when ecologists everywhere became aware of, well, in the ocean but also on land, of the importance of flows of other sorts of materials. And so in the ocean, we learned that nutrient availability varied in various ways across a number of different scales, and that this variation seemed to be important in terms of the ecological response. So the context for this talk is a recent concept that was proposed by Leroux and others in the early 2000s. It's a meta-ecosystem. And that's defined, as I've shown there, it's a set of ecosystems linked by flows of energy, materials, and organisms across ecosystem boundaries. And so if we imagine a coastline, a coastline here, and these little bumps are different rocky shores along the coastline. We'll say they're intertidal sites separated by sandy or cobbly beaches or other areas that aren't inhabitable by the organisms that live in these rocky shores. And there might be some that have a particular kind of community structure compared to others. They are offshore. Oops. You're right, Gary. There are too many buttons. There is a coastal current that you might think might be uniform along the coast, given that it's, in this case, I've drawn it as a relatively linear one. But in fact, sometimes it does vary for various complicated reasons. If we then look at the connections between these local scale intertidal sites, these represent propagules moving from one location to others. Sometimes they move a long distance. Sometimes they actually recruit back to the native site and so forth. But this is one of the major kinds of connections that you see in a meta-ecosystem. And the other connection is one that's delivered, at least in most areas, by the ocean. The delivery of nutrients and detritus might vary along the shore as the current varies in how far offshore it is. So the question of the day, at least that I'm going to ask, is do dynamics of marine meta-ecosystems vary predictably? And I want to emphasize I'm talking about dynamics, so this means rates of inputs, rates of interactions, and so forth. In coastal meta-ecosystems, a key regional scale variable is the regime of alongshore and cross-shelf currents. And before I explain to you what those are, I want to give you the outline of the talk just so you can anticipate what's coming up. I will first summarize coastal current regimes, try to, it's a little oceanography 101, just to make sure that we are all on the same page. I'll then describe the hypothesis that we've proposed to explain patterns of variation in dynamics along the coast. I'll give you the approach that we've used to test it and some data that we've obtained from a variety of systems in these tests. And then I'll conclude talking about possible mechanisms that underlie these linkages. Okay, so coastal current regimes. Basically, we're talking about upwelling and downwelling. And what that means is, for example, an upwelling scenario would look like this. Imagine this is a coastline. Here is the continental shelf. There's usually a broad, flatter part, and then it goes down to deeper water. And in some parts of the world, the California, Oregon, Washington coast is one, southern South America, the west coast of South America. Another, there are 4 to 5 of these areas around the globe. When a differential between the temperature over the land and the ocean sets up during the spring and summer, you have winds that arise and they blow along the coast like this. And as a consequence of this movement, there is also movement of the surface waters away from the coast. This is called the Ekman effect. And what that produces, since water is moving both alongshore and away from the coast, that produces a lowered sea level, which is raised again by deeper water moving from depth to the surface. Deeper water is colder, it's nutrient laden, it has high CO2 and low oxygen. And what happens is eventually is phytoplankton blooms. The opposite condition is what we call downwelling. And notice I'm referring to this condition as persistent upwelling, this to persistent downwelling. I'll explain what that means when I get to this middle part. Downwelling is basically the reversal of the upwelling event. Winds either stop or they sometimes reverse, blowing away from the equator. What this produces is an onshore surface flow, as shown by these arrows here. This onshore surface flow is of warmer water. It's typically lower in nutrients, which have been used up by the phytoplankton in the water. And since they're piling up along the coast, they create a pressure gradient which forces water to depth. So we have sort of a reversing conveyor belt sort of pattern here. The condition in between I call intermittent upwelling, because under some conditions you have switches back and forth between the upwelling state and the downwelling state. And these can occur on a variety of different periodicities, and I'll show you some data that show these 3 different conditions here in a sec. So this is the gradient I'm talking about, this one between persistent upwelling to persistent downwelling with intermittent upwelling as the intermediate state. Okay, so let's give me a little drink. Is this thing working yet? No. Okay, so if we want to look at a gradient of upwelling, the ideal would be to find a coast where you had all of these different conditions that I've just described. Unfortunately, they don't really exist. Along the U.S. West Coast, with Washington up here, here's Oregon, here's California, and then Baja California here. What you have is basically 2 of these 3 states. Most of California has persistent upwelling. The region from about the southern part of Oregon down through Point Conception right here is a strongly upwelled area that's indicated by these cooler temperature colors here along this stretch of coast. And notice how wide it gets, and also how much structure there is in the the currents. So the California current is basically indicated by this wandering line between green and yellow color, or warmer colors. In Oregon and Washington, you have intermittent upwelling. You have a situation where there are relatively frequent switches back and forth, but there's no persistent downwelling. So to get that part of the gradient, we had to go to New Zealand and work there. It was a real hardship, I have to tell you. The South Island of New Zealand is right here, and you can see it's got nice, relatively linear coastlines, sort of like the West Coast. And due to the current patterns that occur there, we have from the west the Tasman current coming in, splitting into two components, one called the Westland current, one called the Southland current. And due to the physics of the situation, the East Coast is a persistent downwelling region, an area where the water tends to be moving shoreward most of the time. And on the West Coast, you have a weakly intermittent upwelling. You can see here this gradient I showed you of temperature here along the coast. You also see here on the West Coast of New Zealand. So this is an area of intermittent upwelling. So we have overlap here between two different regions with the same kind of physical oceanographic conditions, and then we have these two extremes here. Okay, just to show you what persistent and intermittent upwelling and persistent downwelling look like. I've taken some data from a number of the sites that we've used in those regions. The persistent upwelling regime is seen from these data from sites in California, at Cape Mendocino, Kebasilla Hill. These are in Northern California. The next two graphs are from Oregon sites, Fogarty Creek and Strawberry Hill along the Oregon coast. The next two are from New Zealand, Woodpecker Bay and 12 Mile Beach. These are on the west coast, and you can see that they have the same distribution of upwelling. This is the Bakken upwelling index. Higher numbers indicate stronger upwelling, negative numbers indicate downwelling. And this is just proportion of the different categories. And then finally, the two bottom graphs show persistent downwelling, Shag Point and Raramai on the east coast of the South Island of New Zealand. Now notice that when we say persistent downwelling, it doesn't mean that upwelling never occurs. You do have it. These bars right here show that, but it's relatively weak. And also, when you talk about persistent upwelling, you do have occasional downwelling, but it's relatively infrequent. So we're talking about probabilities here more than we are talking about absolutes. Okay, so the intermittent upwelling hypothesis and its predictions, it's actually a fairly simple idea. I have two sets of predictions. One is for the magnitude of upwelling, and the other is for the intermittency, the actual rate at which things switch back and forth. And, you know, ecology is full of intermediate sort of models. Gary talked about some of them. They're very common, and this is a similar sort of idea. Here, the predictions are that the rates of ecological processes, and I'll describe what those are in a second, vary with a gradient of upwelling from persistent downwelling to persistent upwelling, with the intermittent upwelling regime here in the middle. And so it just says that these rates will vary unimodally along this gradient, with high rates occurring during intermittent upwelling regimes and low rates both with persistent upwelling and persistent downwelling, and I'll explain why in a second. With respect to intermittency, this is essentially a measure that tells you what the rate of, or what the frequency of shift is back and forth. The prediction is even simpler, that the rates of ecological processes should just simply increase in some sort of monotonic way against a gradient of upwelling intermittency. And notice that this basically just says that you take this half of the curve and you fold it back on this side. And so we're really looking at increases in both directions from either persistent upwelling or persistent downwelling. Okay, so the predictions, and these are for the magnitude, the unimodal side of the curve. Sorry, I keep getting too close to my mouth here. And we're going to start from the bottom up, basically. Predictions at the lower trophic levels for phytoplankton. I'll work my way up through the higher trophic levels. And this one basically sets sort of the scenario by which other predictions at other levels will follow. For phytoplankton blooms, here is our upwelling gradient. This refers to high and low rates of phytoplankton productivity. And it's a unimodal prediction. And the reason for it with persistent downwelling is basically that with downwelling you get no persistent nutrient inputs. That with persistent downwelling, the surface nutrient poor water is basically always flowing coastward and therefore not able to support blooms of phytoplankton. At the persistent upwelling end of the gradient, there's lots of nutrients, but because of the offshore currents that are continuous, the phytoplankton are carried with the currents and swept offshore and lost to the system. And as a consequence, for organisms that live on the shore or in the shallow subtidal, the access to this resource is relatively minimal. If we go up another level now, looking at larvae and the recruitment of the larvae, again, a unimodal prediction for similar reasons based on this one here, and that is that at persistent downwelling, if you have no blooms, there's relatively little food for the larvae. They shouldn't grow very fast, shouldn't even survive very well, and there won't be that many of them to begin with because of the presumed low productivity on the coast that produces them. At the persistent upwelling end of the gradient, again, these are very tiny, almost particulate things. They will be carried offshore and again lost to the system. And in both of these cases, the intermittent situation simply is one where the water, remember I mentioned the conveyor belt idea, the water is basically being moved back and forth, and so larvae might be swept offshore or phytoplankton might be swept offshore during upwelling, but they'll be returned during downwelling, and if this occurs frequently enough, they will have higher success in getting to the shore. With respect to responses of prey on the shore once they get there, and we're talking about colonization and growth rates, and these are things like barnacles, mussels, other kinds of filter-feeding organisms. At the persistent downwelling end of the gradient, with no phytoplankton blooms and few larvae coming in, there is a low amount of food available, so colonization rates should be slow, and growth rates of things that are on the shore should also be slow. They don't have much food. With persistent upwelling, again, the food is being swept offshore, and so they don't have access to much food. And again, it's this intermediate situation that should bring food regularly back shore, produce food regularly offshore, and then bring it back onshore. And then if we look at the species interactions among the higher levels, looking both at predation effect and predation rate, and I'll define how those differ here in a bit, and competition for space. With persistent downwelling, because there are few prey that have settled on the shore and they don't have much to feed on, there are going to be few of them and they'll be relatively small. With respect to upwelling, you have the same result, but for a different reason. Everything was swept offshore. The densities of the species that might prey on you or that might compete with you are going to be low, and it's at the intermittent stages, the intermediate level of upwelling and downwelling, where you should have the highest population densities. The intermittency predictions are a lot simpler. They look like this, and that's for all of the factors: phytoplankton blooms, larval retention, responses of prey, predation and competition rates, and again, this basically follows this monotonic increasing sort of pattern. Whether or not the response is strictly linear or not doesn't really matter. The prediction is that there's a monotonic increasing effect. Okay, so you might wonder why I'm focusing on upwelling. There are a lot of other things that go on in the ocean. And those include things like evolutionary history, the geomorphology of the coastline, disturbance rates can differ. There are important transport mechanisms like internal waves, tidal bores. River inputs can provide nutrients. Sea breezes are known to cause localized upwelling, and then larval transport and larval behavior can vary. So there are a lot of other factors that could be important. One thing, just to comment, the evolutionary history side of it, one reason we used New Zealand was that even though the species there are totally different from those on the West Coast, they're very much the same kind of animals and plants. But the rationale for using upwelling is that, first of all, we decided to use a hierarchical approach, so we wanted to start with large-scale processes first, and then add complexity as needed or as possible, this kind of complexity. However, it's really not going to be possible for some time because the only really globally comprehensive data set that's available to use in an analysis like this is, in fact, upwelling. We don't have multi-site global sorts of data sets on things like internal waves or tidal bores, larval behavior, etc. So our approach was partly dictated by what was possible. Okay, how do we test this intermittent upwelling hypothesis? First of all, the excuse me, the interaction webs for the two areas are actually quite similar, as I've just sort of alluded to. If you look at the California current, large marine ecosystem, this is the interaction web, so these are the main species involved in the interactions in this system. We have phytoplankton and detritus here providing food for mussels and barnacles that are these things all over the rocks here. And here's a close-up showing mussels and barnacles and one of their major predators, the sea star Pisastrocraceus. And whelks are another major predator. There are whelks here as well. And so this describes a fairly fundamental core of this particular system. On the New Zealand shelf, we have a similar web, but the difference is that there are both crabs and fish that can come into the system from the subtidal. And sorry, and have effects on the mussels and the barnacles, but the main predators still appear to be the sea stars and the whelks. Okay, how do we measure the subsidies, phytoplankton and recruitment? We measure recruitment using the standardized settlement surfaces. This is a scrub pad that you use to wash dishes in your kitchen. Turns out mussels love to settle in these things. And this is a plate with a rugose rubbery surface that's on it that barnacles like to settle in as well. Here's a settling stage, a cyprid of barnacles, and here's a metamorph, and these are easily attracted to these surfaces and then counted. The way we measure phytoplankton and nutrients is basically just go to the edge of the shore, dip a pole in, and collect water in bottles, and then process it in the lab. And these things, by the way, we've done these for as many as 24 years at some sites. So we have a long-term data set for both the recruitment time series and the phytoplankton. In terms of prey response, we also for a long time have been measuring mussel growth rate. And the way we do this is we take mussels from the shore, we put them into plots, put some mesh over the top like you can see here, let them reattach, and then remove the mesh, and then a year later we come back, remove the mussels, measure their growth from the initial length, and do this repeatedly from one year to the next at a whole bunch of different sites. For the barnacle colonization part, in some of our experiments, and I'll show you some of these in a second, what we do is we clear the rock and then we follow the colonization patterns, and barnacles typically show up first. We follow the colonization patterns through time on a regular basis, so we can get increases in abundance, increases in size if we want to from that. Field experiments to measure are used to measure predation effect and competition and colonization rate, and I'll show you the predation rate over here in a second. But the predation effect is measured by using fairly standard sorts of experiments. These are just like the ones that Joe Connell did way back in the 50s. There is a stainless steel mesh cage that keeps predators out. And again, we're talking about creeping, slow-moving predators like sea stars and whelks. There is a control for the mesh of the cage. Here it's a roof, so that's basically a two-sided cage which allows the predators to get underneath it. And then there's a marked plot, which is free of any of the artifice of these devices used to manipulate things. And so you can, by looking at the results in the predator exclusion, comparing to either of these two different treatments, you can get an idea of what effect the predator has on the abundance of prey in these areas that they're colonizing. And so you get from this a colonization rate as well. And then you can get a competition rate between mussels and barnacles and whatever else comes in by looking at the events that occur in the cage. And I'll show you a diagram of how we measure rates in a second. Predation rate is defined differently from predation effect. Predation effect looks at the effect of predators on things colonizing into the plots. Predation rate starts with a fixed number of prey, and here we have a clump of 50 mussels, another clump of 50 mussels, and what we do is we look at the rate of loss of these things in the presence and absence of predators. So the fence excludes sea stars and whelks. The partial fence is a control for the mesh, but it allows predators to enter and obtain and eat the mussels, and well, in this case, the mussels. Okay, so quantifying species interaction rates we use these data, and so to quantify predation effect, what we do is we look at prey abundance first through time in these experiments, and we use total prey abundance curves, which is what's shown here by this dotted line and this black line. They differ because one is the curve in the presence of predators and one is the curve in the absence. we look at the effect in two different periods of time. One is on the colonization rate of prey, and that's simply the difference between the slope of the lines to the point of inflection here of the prey curve in the presence of predators compared to the slope of the line at the same point in the the prey curve with predators absent. And then we look at the effect on the final prey abundance by just looking at the abundance difference in prey between the two different experiments, the cage versus, in most cases, we use the roof as our control. To calculate competition and colonization rates, we use just the results from the cages. And in this case, what you typically see is that barnacles will colonize first, and then mussels will come in and displace them, overgrow them, smother them, and outcompete them. And so we measured for barnacles two different rates. One, the colonization rate was simply the rate at which the abundance increased in the first part of the experiment, and then the rate of predation of competition with mussels was defined as the negative slope of the decline in abundance of barnacles as they got overgrown by mussels. Okay, so I'm now going to show you results from the data, and I'm going to show you results for the intermittency and for the magnitude of upwelling. And so in these graphs that I'm going to show you, I'll first show you the magnitude predictions. They're going to be data points for 3 different locations. The red will be for all the New Zealand sites, the blue will be from Oregon sites, and the green will be from California sites. So this is looking at the ecological subsidy results for phytoplankton here, barnacle recruitment here, and mussel recruitment here. Here's the upwelling gradient, again using the Bakken upwelling index. And you can see that in all cases the curve is unimodal. That was as predicted, that the different regimes separate out as expected. That is the green California persistent upwelling sites are all here to the right. The downwelling sites in New Zealand are all here to the left. And then the Oregon and west coast of New Zealand sites that are intermittent are all sort of clustered together with the New Zealand sites a little bit to the left of the Oregon sites. But also notice that there's overlap between the different point clusters, which suggests that these are responding, the ones in New Zealand and the west coast of the U.S. are responding similarly to these different oceanographic conditions, even though they're in totally different parts of the world. Okay, the prey responses, again looking at magnitude predictions. Mussel growth and barnacle colonization rates are also unimodal, with similar sorts of patterns as I mentioned in the last with the last slide. And one interesting thing here is that the mussel growth response is similar despite the different mussel species that we had to use. As I said, there are virtually no species that are shared between these two regions. So the dominant mussel along the west coast of the U.S. is the California mussel. There is an ecological equivalent in New Zealand called Perna canaliculus. Same size, grows very fast, dominates space. So these are ecologically equivalent species that are responding in a similar sort of way in their different locations. And then looking at species interactions, looking at predation rate, predation effect on the colonization of prey, predation effect on the final abundance of prey, and then the competition rate. They're all unimodal. Some are weaker relationships than others, but the pattern is pretty consistent. Okay, so let's move on. These are intermittency predictions now. Remember, the prediction there was for some sort of monotonic relationship. And you can see here for the subsidies, the ecological subsidies, that only one of them is monotonic. That's for the mussel recruitment. The pattern for barnacle recruitment and phytoplankton actually were unimodal. And the South Island of New Zealand West Coast sites are generally separated from the, see, do I have little circles around them? Yep, I do. Are generally distinct from the other regions. So these cluster together and don't really overlap too much, at least in these graphs, with the other sorts of sites. And then one point to point out is that mussels are the linear one, barnacles are responding somewhat differently, which is actually consistent with what we found with them, that both these species in the larger sense tend to do things kind of similarly, but if you look at them on a finer scale, barnacles and mussels do their own thing. Prey responses, mussel growth on the top, barnacle, barnacle colonization on the bottom. One of the two is monotonic, the other one is not. Species interactions, in this case, only one is not unimodal, and that's this one here, which is a saturating sort of response. This is the predation effect on, there we go, predation effect on final prey abundance. Otherwise, competition rate and predation effect on colonization are unimodal. Notice also that with all of the intermittency predictions that the R-squareds tend to be somewhat lower. They don't explain as much of the variance. So in the case of the intermittency predictions, only 3 of the cases are monotonic, as is predicted by the model, and so the question is why? In looking at the data, I noted that the New Zealand West Coast sites tend to have higher intermittency than the Oregon sites. That is, the rate at which they switch back and forth between upwelling and downwelling appears to be faster. So that suggests that the idea that the New Zealand West Coast sites might be over-intermittent. That is, they switch back and forth between upwelling and downwelling too frequently for the system to be able to respond in the same way that it does on the U.S. West Coast. And as, just to give you a sort of a time frame, it takes about 3 days for a phytoplankton bloom to occur after upwelling starts. So there's a lag in there that has potentially important effects. So we went to the Bakken index data sets to look at the duration of upwelling events and calculated how long they were in Oregon and on the west coast of New Zealand. And in fact, the length of the upwelling events is shorter, substantially shorter, on the west coast of New Zealand than along the Oregon coast, which is the other intermittent site. So this might be supportive of the idea that the system periodicity in New Zealand is just too frequent for the kinds of responses that we expected to see to occur. Okay, to summarize this part of the talk, the unimodal patterns were seen in all cases with respect to the magnitude measures. And if you average across all of those data, about 53% of the variance was explained by coastal oceanography, by the upwelling regime. With respect to the intermittency predictions, only 6 of 9 were not supportive of the monotonic increasing pattern, and they explain about 37% of the variance, whether they're monotonic or unimodal. So I've already suggested this, that the possible explanation for these differences might be this over-intermittency idea, but it would certainly could stand some more research. Okay, so this brings me to the final part of the talk, which is, let's see how we're doing on time here, the mechanisms that might underlie these linkages. And what I'm going to show you is all correlation analyses. I mean, we've done a lot of work on mechanisms, but with respect to the various linkages between these things, at the moment, the best we can do is look at the relationships between the data in these data sets. A little background to talk about what things you might expect with the influence of the lower trophic levels and how they flow up to the higher trophic levels. I'm going to first summarize, and I'm sure all seen this, the information from studies by Fretwell and Oksanen proposing a bottom-up, top-down theory in which they suggested, building on the Harston, Smith, and Slobodkin ideas, that as you went from a region of low productivity or low nutrients to a region of high productivity, so here's the gradient, that you will see successively along this a place where plants are the dominant organism, and they're very abundant because they have no consumers that are influencing them. Eventually, productivity increases to the stage where herbivores can invade and become abundant and start having an impact, and they will reduce the abundance of plants. If you have further productivity, then carnivores will supposedly invade, begin to reduce the abundance of herbivores, which should free up the plants from herbivore control and allow them to become abundant. And of course, this is the HSS prediction right here. And then with further productivity, you could go on it forever on this, but typically it's thought that 4, maybe even 5 levels is about the maximum there will be. And this sort of alternation of control is seen in a lot of different places and certainly has a good amount of empirical support. But there also are situations where it doesn't work. And an alternative way of looking at it is a simpler one in which bottom-up effects drive top-down effects, and omnivory occurs and blurs the trophic structure. So you might envision 2 states, one where bottom-up effects are low, and so I'm using, again, a upwelling gradient here, but this could be productivity or nutrients. Where it's low, you're going to have low production of detritus, low production of propagules, low abundances of prey as a result, low abundances of predator as a result of that, and minimal effects on the prey because the predators never get very abundant. And that could contrast to this strongly bottom-up influence system where these factors are all important. The biomasses are high, the flows are high, and so forth, and predators can have a strong effect on their prey. The predictions from this model are quite simple. Again, they're monotonic increasing that predicts that inputs from the lower trophic levels will lead to increasing inputs from the lower trophic levels will lead to increasing responses by the higher trophic levels. Okay, so given that as a background, what do we see in relationships between phytoplankton measured as chlorophyll A versus these higher level processed rates? And so I've got here the predation effect on the final abundance of prey, predation rate, competition rate, barnacle colonization rate, and predation effect on colonization. And you can see in all cases that the relationships are monotonic increasing. The more phytoplankton production, the stronger the interactions, rates of the interactions. With barnacle recruitment, sort of another level up, versus these higher level effects, again, we have mostly monotonic effects, except for the effect of predation on the final abundance of prey. And in this particular case, at the highest levels of barnacle recruitment, the final effect of predation is actually lower than it is over here, which might mean that very high barnacle recruitment, way out here, overwhelms predators. In other words, they just can't keep up with the input of prey, and that reduces their impact. Looking at mussel recruitment versus higher level rates, here we see only one of the 5 responses that is monotonic increasing. The others are all unimodal. And I think this might be driven by low barnacle colonization rates on the west coast of New Zealand, which is what I've circled here. These are the sites on the west coast. And these are low rates, even though mussel recruitment as measured in these toughies is high. And this is likely to feed into all of these other things. Barnacle colonization rate will affect competition rate. It'll affect the predation effect on colonization, and it'll affect the predation effect on the final abundance. Barnacle colonization rates versus higher level processes, these are all monotonic or saturating, and then the same with species interactions related to competition affecting predation, predation effect on colonization versus final abundance, and predation rates versus the two different kinds of predation effects. Okay, so to summarize then, as predicted, the subsidies, the responses, and the interactions all varied unimodally with upwelling magnitude, but not necessarily intermittency. And this explained about 50% of the variance in these different rates. I think this is actually kind of a remarkable result. I actually never expected the coastal oceanography to have as big an effect as it apparently has. I knew there was an effect because we've been working in this system for a long time, but to explain 50% I think is kind of surprising. But there is another 50% that isn't explained, and so what explains that? And I think that's where these more regional to local scale sorts of factors come into play, and also things like evolutionary history. So there are a set of factors in each one of these categories, all of which might have an influence. The lack of conformity of the data to the intermittency predictions might lead to this, an examination of this idea of over-intermittency. With respect to the mechanisms, 17 of the 21 comparisons were consistent with a bottom-up forcing sort of hypothesis by subsidies or by lower level factors on higher level factors. So this is probably one of the big questions. Where does the model apply? Now, I've used it for two sites in the same major ocean basin, the Pacific Ocean. But I'd like to suggest that it actually might apply to most coastal regions around the world because with the exception of things like embayments and other irregularities along the coast, most regions do experience a regime of upwelling and downwelling, with one of these dominating in some places and the other in other places. And this varies. And so it seems likely that these predictions might be applicable more widely. Another implication, which is a more pragmatic one and gets to what might be happening in the future in this system, is that there might be a biogeographic shift, at least in the dynamics of the system. So here's the model. Here's what we see now, at least on the west coast. Oregon is clearly in the middle, where this intermittent upwelling is a really strong factor. California sites were all consistently over here. There is a prediction made about 23 years ago by Andrew Bakken, the man who invented this upwelling index, that with increasing climate change, increasing temperature differential between continental masses and the nearby ocean, that upwelling is going to intensify. And in fact, some recent work by one of my students has shown that it actually has intensified over the past 30 to 40 years. So we know that it's increasing and that it's likely to continue increasing. So the consequence of that could be something like this, where Oregon might grow to resemble, at least in terms of the dynamics, grow to resemble California, with weakening of a lot of these rate processes that I've discussed with you. And then California is going to become even more like California, according to this kind of model. Future directions. One thing I'd like to do would be to test this using data from other systems. I know there are some data available in Chile that might be useful. Other regions would be interesting as well. It'd be nice for somebody else to do these kinds of things as opposed to me, so there's some kind of Menge independent testing going on. The mechanisms, I've already said, these were mostly correlations. It would be useful to look more strongly into the mechanisms that underlie these relationships, and also to get at factors that might explain some of this 50% or so of the variance that's not explained. One thing I haven't pointed out yet, and I will now, is that most of what I talked about has been with the invertebrate-dominated part of the intertidal food web, but macrophytes are algae and seagrasses and so forth are a major part of these systems. And there may be reasons to think that they respond somewhat differently, since you might expect with increasing nutrients that macrophytes would just simply steadily become more abundant. That could lead to a different dynamic at the persistent upwelling end of the gradient. Another issue relating more to what Gary was talking about was how does community structure vary with upwelling? We have the data to look at these things, diversity and abundance, and that will be a next step in what we do. My prediction is that the relationships are going to be a lot weaker, that we're not going to see as strong a relationship between an upwelling gradient and diversity and abundance, simply because species interactions tend to block some of the changes as you get into the more complex top part of the food webs. Another issue is where are the nutrients coming from in these downwelling dominated places? The east coast of New Zealand has, as you saw, persistent downwelling, but there actually are high biomasses of seaweeds there. What's supporting them? Part of the story is probably that these are able to store nutrients when they do become available. Another part of the story might be that river inputs are a big source of nutrients for those kinds of regions. And I'm done. This is my granddaughter, who is gleefully running away from her mommy and is currently the light of my life. That's her right there. These are my acknowledgments, and thank you.
I think we have time for one quick question.
Hi, great talk. So you talked about the impacts of climate change and potentially intensifying upwelling and how that might affect. I wonder if you have any insights from the impacts of, say, the Pacific Decadal Oscillation and ENSO on your data, and how that may change with climate change and what that might play into.
Yeah, we've analyzed that, looking at ENSO effects on the growth of mussels and on phytoplankton blooms, and comparing it to the effect of the PDO. And it turns out ENSO has a big effect on both phytoplankton bloom magnitude and also on mussel growth rates. The effects of the PDO, the ENSO, and another cycle called NPGO, North Pacific Gyre Oscillation, were looked at with respect to recruitment of barnacles and mussels. And they have an effect. The biggest effect is actually of the NPGO, which is like a 10 to 15 year cycle in which currents vary in their magnitude. So there are definite links. And the important thing, I think, is that all these climate cycles actually mimic some of the things that are going to happen as the climate changes and changes upwelling regimes and current regimes and so forth. So where we are now is trying to figure out what impacts these changes are going to have as they flow up through the food webs in these different locations. We have some results from a more recent paper that suggests that interactions can, as I've implied here toward the end, block some possible impacts of climate change. So, for example, barnacle recruitment is necessary for mussel recruitment and growth, but barnacles don't seem to be responding to climate. Mussels do. And so it might be that no matter how abundant mussel recruitment gets, that if the barnacle recruitment rates don't change along this unimodal sort of gradient, that there will be no response by the mussel population because they're totally dependent on this facilitation by barnacles. So that's just an example of the kinds of complications that I think are going to be very hard to take into account as we try to predict what these systems are going to look like in 50 years.
Okay, I think we'll have to hold any further questions until the panel discussion. Let's thank Dr. Menge one last time.